So let me just introduce Jo quickly. Jo is the senior curator of algae at the Natural History Museum in London. And in this role, she is responsible for the management and development of the NHM algal collections. At approximately 400,000 specimens, this is one of the largest collections of its kind in the world, and contains specimens from a diverse group of organisms like seaweeds, freshwater algae, and cyanobacteria. Jo's research interests are focused on the taxonomy, biodiversity, and conservation of freshwater algae, seaweeds, and bryophytes. I won't take any more of your time. Jo, please go ahead. And if you share your screen, we should all be able to see it.
Okay, so does that come up?
Yeah.
Okay, great.
Yes.
All right, super. So, okay, so I'm going to talk about Desmid algae this morning. So, the kind of typical Desmid algae, if you have some idea what these organisms are, this is an example of a few different genera that are kind of, typical Desmids really. They're quite striking, microscopic green plants. So when they were first observed under the microscope, people thought these were actually two cells joined together, and that's where the term Desmid comes from, because the Greek desmos means bond or chain, as in two cells joined together. And so subsequent investigations found that these are actually one-celled organisms with two semicells, so they're symmetrical half cells. Obviously the name has remained the Desmids, and they're beautiful organisms. They're microscopic. I suppose the largest, just to give you an idea of scale, the largest of these little mini plants would be maybe up to 1/3 of a millimetre. So you can just about see some of these with the naked eye if they're kind of right, if you've got them in a little watch glass. But really they are microscopic, but very beautiful and fascinating organisms. Although they're unicellular, single-celled organisms, they can form loose associated chains. So you can get filamentous forms like this Desmidium, but they're not true filaments. These individual cells will break apart very easily. But there's an amazing variety of forms and structures in this group. So I think we'll talk a bit about the biology of this group, exactly what they are, so you can get an idea of what the algae, the Desmid algae, actually are. But first of all, where do they live? Where do you find these? Well, you can find Desmids pretty much all over the place in freshwater habitats, but I suppose they're classic diversity hotspot kind of habitat would be these extensive bogland, peatland areas. This is a image up in northwest Scotland of beautiful boglands, and if you're tramping around in this landscape, then you can see there's lots of little pools fringed by sphagnum mosses, and if you peer into these sphagnum pools, then you can see a variety of micro habitats within here. You've got your feathery sphagnum, pondweed, things like Utricularia, the carnivorous pondweed are all super habitats for these Desmid algae to kind of loosely associate with. And if you took a clump of sphagnum or pondweed and you kind of gently squoze it into a little jar of water and let it settle, then if you're in a Desmid rich habitat, you would see, once it's settled, these green clouds of Desmids have started to rise up from the bottom of the jar. And if you put one drop of this water onto microscope slide, then if you're really lucky, you'll see a whole slide full of these bright green little one-celled Desmid algae. And I put that series of slides in for two reasons, really. One point to make is if you can see from I've got a lot of sediment and sludge in the bottom of that jar, but if you're in a desmid rich habitat, then these organisms, although you can't see them when you're tramping around, they're actually forming quite a considerable biomass in the environment. So they can have, quite an impact. There's a lot of them. The mass of this group must be quite considerable if you kind of look on landscape scale. And also the other point is that beautiful landscape that I showed in the first image there. If you wanted to survey the Desmids, then you're looking at one drop of water at a time. It's trying to do a microscopic survey on a landscape scale has got a lot of issues to try and work out what's there. And that's something that we'll come back to again, perhaps at the end of the talk. So a little bit of biology really about the Desmids. So what are they, what defines this group? Well, they belong to a group of green algae called the conjugating green algae. And basically that's referring to the unique sexual reproduction of this particular group of algae. They don't form flagellated gametes. They basically have the two cells of the two organisms will sidle up to each other, and the cell contents kind of go into an amoeboid-like state, and they'll join together, and the cell contents will fuse and form a zygospore, which is the sexual fusion of these two individuals. And the Desmids, they can be quite ornate. So that big spiky ball in the middle is a zygospore of this microsterious species. And you can see the two empty cells either side of the zygospore from where the cell contents of in amoeboid fashion moved out and fused together. And the zygospore is the diploid phase of this life cycle as well. So the vegetative, the green cells that you see floating around are haploid, which means they have half the number of chromosomes, and the diploid phase has got, your two chromosome pairs together. But most of the time, the Desmids can't be bothered with sexual reproduction. It's just too much hassle. So they usually just reproduce vegetatively. And there's a beautiful picture here from, this is the Nikon Small World competition. So this is showing the microsterious, which is the same species as a previous, same genus as a previous slide, how that will divide, and now the nucleus in the center of the cell will divide, and then the cell will start to form two new semicells, which will then expand, and then you'll get the two cells joined together, and then they'll float off. So it's quite interesting, really, if you're looking at one of these single celled with the two semicells. Often, if it's a mature specimen, and the two halves of the cells are quite likely to be different generations. So although they look pretty much the same, they might actually be different ages, which I think is quite interesting. And I said before, so they don't reproduce sexually very often. It's quite rare. Often be triggered by environmental factors, perhaps stresses in the environment. So usually vegetative reproduction. The significance of them being haploid, half the chromosome component in the main vegetative phase. The reason why I mentioned that is that if you have a mutation then in these cells, it will be more immediately expressed. And I think that's the reproductive biology has a bit of an influence on the taxa that we can see. So there are a lot of Desmids been recorded in the UK and Ireland, and overall in the world, I think there's about 6,000 of these organisms, species and subspecies have been described. So, but only in about 30 in the UK. That's in about 33 genera. So you've got these huge genera with lots and lots of species with very discrete small differences. And they're living in these isolated pools, they're reproducing vegetatively with occasional mutations. You can kind of start to get a feel of how these organisms have evolved, and why we have the number of described taxa that we have at the moment. So the biology does have a kickback into this taxonomy of this group as well. And the striking thing about the Desmids that people would immediately notice is the symmetry with these semi-cells, particularly in this main group where you've got these very distinct semi-cell species. So things like the Cosmarium, you've got, if you tip that Desmid on its head, which this is one of Chris Carter's photos, you can see it elliptical in outline. So it's the Desmids are very much three-dimensional objects, and when you're just looking at them on the microscope slide, particularly with the optics involved in that, it's very easy to get into the mindset of these being just two-dimensional objects, but they're not. They're three-dimensional, and it's very important to see around the whole object, as it were, when you're trying to work out what you've got on your microscope slide. And if you look at something like a starastrum, if you tip the starastrum onto its head, looking down the microscope, then you see it's triangular in outline, so you've got this triradiate symmetry, and you can get much more complicated, symmetries involved as well, which are part of trying to work out the organisms. And the other fascinating thing with the Desmids, the chloroplasts, and they have the most diverse, largest and complex chloroplasts perhaps in the whole of the green plant lineage. And on the left there you've got a genus called Netrium, and it's just got an amazing structure. So if you tipped that Desmid, if it was, if it was as large as a telescope and you have to hold it up to your eye and look down the middle, then that's the circular diagram at the top there. That's what that's showing you looking down. And the shaded part is the chloroplast. So you've got this chloroplast core, and then it's radiating out with all these notches. So they're incredibly complex. And the chloroplasts are also very key in trying to identify these different organisms as well. So quite a key character. And another important biological feature of the Desmids that's worth mentioning is the huge amounts of mucilage that some of the representatives of this group can generate. So this is a Hylotheca. It's one of the Desmids that can loosely associate themselves into chains. And you can see in this picture again from Chris, he's put a blob of India ink onto the slide, so which is repelled by the mucousy, otherwise transparent see-through slime that is produced, and you can see the extent of that around the cell. And a lot of the Desmids do this. In fact, a lot of the algae in general produce various slimes. It's one of their things, and there's a lot of reasons why this is quite a useful attribute for them to have. In the case of Hylotheca, you'll often see it in gelatinous clouds floating in little pools. So you've got a kind of buoyancy aid factor there. But then there's a whole range of reasons why there might be advantages to having this mucus. Whether it's trapping nutrients, so they're close to the cell to absorb, protection against desiccation would be another key issue, and also the impact on the landscape as well that all this kind of goo is going to be fairly considerable in other kinds of landscapes. Mudflats, for example, which are not a Desmid habitat, but other microalgae producing mucilaginous substances can help bind mudflat surfaces together. So there is an impact from this, and also potential food source as well for other organisms. So if we just skip back to looking at the conjugating green algae, so we've looked at some of the key characteristics. So we've got an idea what this group is. We've got the two main groups of Desmids. So within the conjugating green algae, you've got these the so-called sacchaderm or false Desmids, which are forming one taxonomic group in the classification, and then you've got another group of placoderm or true Desmids. And within the conjugating green algae, there's also the truly filamentous algae like the Spirogyra, which some of you might have come across. It's kind of a classic school, what used to be school microscope feature where the cells are so large and you've got the chloroplast spiraling. But the Sacchaderm false desmids are actually more closely related to some of these filamentous algae than they are to the true desmids. But I would say that the classification of this group is still not particularly stable, and you know, I expect there'll be quite a few changes as more molecular work gives us more evidence and insight into this group. But one of the distinguishing features which separates a lot of these groups are the cell wall structures. So you've got that Zygomatales with the smooth walls, and then the true desmids have got slightly more complicated cell wall structures. We've got pores, cell wall ornamentation, and such like. So there are differences, and I thought we'd just kind of run through some examples of some of these groups, just so you can get the idea of the variety of these Desmid algae. So the family Mesoteniaceae, which is those false desmids that were mentioned, got very simple structures, simple cell walls. As you can see here, the chloroplast, again, you've got the we've got the circle view underneath that cylindrocystis. That's as if you're looking straight through the cell. So it's got a slightly stellate chloroplast, rather versus this Spiratina, where you've got a flat ribbon-like chloroplast spiraling around the cell. So just an example of how key these features are in identifying. You'll find things like the mesoteniums, they'll often be in subaerial habitats. So not all desmids reside only in beautiful bogland. A lot of this group will often grow in over wet bryophytes or dripping rocks, and they'll kind of form a gelatinous mass, and that's again where the mucilage is coming in as a desiccation protection for this group. In fact, they can occasionally form a bit of a nuisance actually when they really excel in numbers, and you'll get this smothering effect over bryophytes. And then in the true desmids, the placoderm desmids, you're starting to get more complicated cell wall structures. So you're getting pores and ridges, and there's a few genera that are just included in these three families. Here, got a couple of examples with Closterium, the green bendy bananas, and Penium, both very common genera. But then the Desmidiaceae is the group where many more of the genera are grouped together, and these have got much more complicated cell wall, pores, and as you can see, it's a huge diversity of different structures that would come into this group. So it's probably where you'd find most. And also you've got your the more typical desmids, so to speak, where you can see that constriction in the cell wall. So you've got like a little waist in the cell and the two mirrored halves. But again, they're an extremely diverse group. Desmids in Britain Island, a huge number of species have been described, and there's been a long history of study as well. I mean, these are very beautiful organisms, so early microscopists took a particular interest in the desmids. They're a father and son team in the late 1800s to early 20th century, George and William West, who wrote the Desmid Flora for the British Isles, did a huge amount of work on this group as well as other algae, described many species and varieties. So there's been a long history of study of the desmids. So there's a huge number of described species in Britain and Ireland. The checklist probably about 1,350 species and varieties and subspecies have been described, but we don't really, despite this long history of work, we don't really have much data on the occurrence of these species. So the distribution data, the data that you really need in order to make assessments about the conservation status of this group, and desmids, those restricted to fresh waters. Fresh waters are one of the most threatened habitat in the world. And the desmids are very sensitive to water pollution. They're often used as indicators, in fact, of water quality. So it's really important to have an idea where they occur, are they threatened, are they disappearing, you know, what factors are affecting their population. But we to do usual conservation assessments, your IUCN Red List, you need that data about where the extent, where does this species occur, is it declined in population or increasing. So there's a huge amount of data that is lacking really for us to make assessments about this group, which is a problem really, because microorganisms, it's easy to forget about them because they've not got feathers and fur, and you can't see them without a microscope particularly well. But despite being microscopic, they've got a huge impact on environments and landscapes, and they can be as threatened as the multicellular relatives. There has been an initial review of the Desmid algae back in 2007 for a plant life report, where the colleagues used data from national experts in Desmid taxonomy, Mr. David Williamson, who's worked on this group for over 50 years in the UK and amassed a lot of paper records from his notes of where he's found different species. So there was some initial work done to indicate where important areas for these Desmids are around the UK. And it was also nice to see in the big report that's come out last month or so, coordinated by Kew Gardens on the state of the world's plants and fungi, that we were able to include algae in that group as well. I mean, albeit we were mainly pointing out how much we didn't know about a lot of these groups, but sometimes it's important to make those statements as well. So, we're not just kind of sweeping them under the carpet, so to speak. So we had a project recently at the Natural History Museum with colleagues where we're trying to improve the data set that we have for the Desmids, so that we can perhaps do a little bit more work to look at the conservation assessments of this group. And what we did a couple of years ago, and we pretty much just finished this now, is to digitize all the records and paper-based work that Dave Williamson has generated over the last 50 odd years, and to get that into database so that we can interrogate it a little bit more efficiently, and that accumulated in around 18,000 records for these species. And this map here is just showing where David had identified Desmid samples across the UK and Ireland. So obviously this is a limited approach, but extremely extensive, considering it's really one person's effort. If you just switch that map to show the number of species recorded at the different sites, then you can see those hotspots coming up for the English Lake District, North Scotland, Western Ireland, although Highland's very under-recorded, and I suspect North Wales is as well. But it gives an idea. You can see that Atlantic bias for where which is correlating with those kind of boggy peatland areas. You can also indicate David's home there in the middle of England, under his own little hotspot, which is quite nice, because you can get an idea of what the background, if you were to sample extensively over the whole of England, perhaps that would be a background indicator showing that Desmids can be found all over. They're not restricted to these beautiful northwest habitats. And one thing we found when we were doing this work to put David's records, digitally, so that we could, build up a useful data set to share with the community and this conservation assessment work from, was that there was quite a large disparity between the checklist of species and subspecific taxa that we had for the UK and the list of taxa that David had been recording. I think over his many years and very intensive work, he'd recorded about 800 taxa, and yet for our UK list, we had, it had kind of got up to about 1,350. So there was something not quite right. So we've been going back to the checklist to try and go through that a little bit more critically. And sure, there are some nomenclatural changes where, you know, you can kind of get a little bit more alignment, but the fact remains that there's a huge number of species that have been recorded for Britain and Ireland, but have just not been seen since they were first recorded, perhaps by William and George West back in 1900 or so. And I think another thing that came to light very clearly was that a lot of Desmid taxa are just very rare. And if we go back to our thoughts earlier in the talk about, you know, their reproduction biology, the habitats that they live in, and how, you know, we've got a lot of very similar morphological species, subspecies units that proliferate and stay in these stable populations. We kind of get an idea of how a lot of these taxa are just very rare and live in particular places, and also the difficulty as well of surveying these things and knowing that you've captured a good impression of what diversity is occurring in a particular area. And I've just put this up as an example. Lovely tarn up in the Lake District, English Lake District, School Knot Tarn, and there's over 100 Desmid taxa recorded from there. So it's a super rich site, and there was a fairly intensive survey in 2017, and there'd already been several other visits by David, for example, David Williamson, in the past. But the 2017 survey only found, you know, there were only about 10% of the taxa that had been recorded in earlier surveys. So you think, well, does that mean has the flora of this Desmid community changed over that time? Or, you know, because these organisms, these microscopic plants, can be associated with very specific microhabitats within this landscape, is the sampling just, you know, completely off? Is it just too random? Every time you go, you're going to find a completely different community. So, and a similar phenomenon really is found by my colleague David John, who's done a lot of work in Western Ireland, very intensive surveying, did a few years ago for publication. And the West's father and son team back in 1900 something had done a lot of work as well in Ireland, and David John was finding that, you know, he wasn't really finding species that they had recorded. And, you know, you think, well, is that because they're not there anymore? And we know there's a lot of pressure on freshwater systems, and we know that these are very sensitive to pressures. So, you know, perhaps they are disappearing, or is it something a little bit more niche with the populations and how they change over time? So we've got a lot of questions that we don't know, and a lot comes back to the difficulties of doing a landscape, you know, doing a landscape survey at a microscopic level. And something that we're quite interested in you know, in taking forward, you know, the next steps is looking at how the DNA can really help unravel or give us some extra information about a lot of these processes. So these are not new techniques, but we're finding a lot of techniques in DNA analysis, although they might not be new, they suddenly become cheaper to apply, and then they're a little bit more commonly used. For example, environmental DNA is something that has been used for a while. People are using that, for example, survey for things like, you know, these multicellular animals that people are so interested in, like newts. And so if a newt swims through a pond and it sheds mucus and poop or whatever else, you can take a sample of that water, and the environmental DNA from the newt in very small quantities will be present, and you can magnify that and say, okay, you know, this is present in here. And similarly, you know, in other similar techniques where you can also look at the metabarcoding. You know, this next generation sequencing is basically rapid magnification of DNA strands. so that you can get huge amounts of data. You know, it gets so complicated. You need to work with specialised bioinformaticians just to make sense of the data that you're generating. But you can do a huge amount of interesting stuff with looking at the DNA, and put another example up here of the diatom colleagues who have been using a similar approach to try and improve their surveying for the Water Framework Directive, for example. One of the problems, though, with applying this to something like the Desmids is that you do really need a reference library, so you can find, you know, DNA in your water samples, but really you need to be able to reference that to a taxon, you know, an idea of the voucher specimen, so you can say, okay, this is matching to this, I've got a nice DNA library. And that's something that we don't really have at the moment for the Desmids. For the diatoms, there's been some work already started to create such a library. It's very slow, it's difficult. You often have to culture the material, which is time consuming and resource costly. But there's definitely a lot of interesting avenues there that, in partnership with the traditional techniques, you're still going to need your microscope. We can perhaps, you know, get a real, you know, a better idea really about what's going on with these Desmid populations, and have information that we can then really feed into our conservation assessments of this group, because we don't really know what's going on with these. Taxa could just be disappearing, you know, and we wouldn't really know at the moment. And I'm really keen that, you know, we don't just forget about these microscopic plants just because we can't see them. And I'm hoping that, you know, we can get more data together, working in partnerships, and actually get them, you know, as up there and noticed as a great crested newt. So that's basically the end of my talk about Desmids there, and I've just put a few acknowledgements up for some of the colleagues that I've been working with on these projects, like Dave Williamson, a national expert on the Desmids, and some of the colleagues. And a lot of the pictures in my talk were with Chris Carter's picture up there, and we have a photo catalogue project, Algae Vision. So if you're interested in Desmids, you can take a look at that and see some of our other images. So there we go. And if anyone's got any questions, I'll have a go.
Thank you so much, Jo. That was absolutely brilliant. We our Q&A box is popping, so let me just go right in.
Can I?
Yes.
Will you relate the question?
Yeah.
Right, okay.
Do you want that, or you can take a look at them in the Q&A box, whatever is easier for you.
Hang on a minute. Let's see if I go. Okay. So if I go back to. Shall I stop?
Okay, so I'll just ask the questions if that's okay.
Yeah,
okay.
Should we do that?
Yeah. So Lauren Gardiner has a question. Can you tell us more about how Desmids are recorded and preserved in natural history collections?
Well, it's a good question. I'll just pop that over there. It's not very well, I suppose, would be the short answer. They are problematic to preserve, and it is a great personal sadness that is the case. I mean, I used to work on, well, I still do work on bryophytes, mosses and liverworts, and they're so easy to preserve. Put a moss in a paper packet, leave it in your coat pocket for 3 years, forget about it, and you can rehydrate that specimen and the cells look alive. But the problem with the Desmids is a lot of the characters for this group that you need, particularly for those false Desmids that I mentioned, are relating to the chloroplast. So, and that the chloroplast will often shrivel up or change shape if you preserve them. I mean, the best way to preserve the Desmid samples is probably going to be is in liquid, a preservative liquid. Traditionally, people have used formalin, and nowadays they're probably more likely use something like glutaraldehyde, so preservatives basically. But it's not ideal because you you've kind of got then a community of you've not just got one isolated cell, you've got a community sample that you've preserved. So you might find one cell of a particularly, you know, a species of interest, and then you presume there's going to be other cells of that in your sample, but it's not quite as satisfactory as having a herbarium sheet with a, you know, your flower stuck in the middle. Interestingly, we do have historic samples at the museum from the 1800s, really, where they did just preserve the specimens dried onto sheets. So they'd dry a little bit out, sometimes on a piece of mica. So you just have a little bit of dry sample, really. Just, you know, so it's on a piece of paper, put your drop of water on a piece of paper, it just dries out, and then put it in a packet. So in the 1800s, that's what they would do. And I think you'd kind of, you know, I think they have been slightly dismissed, although last year we did have a little experiment where we rehydrated a little bit of this old paper from, you know, late 1800s, I guess, and we're able to pick off a desmid and put it under the scanning electron microscope, and you could see the cell wall structures. And so that was another angle for the, you know, for the 101 projects that you don't have time to do. This on the list is to try and look at some of our historic material and to see, using the SEM, if we can actually get, because the main problem is the type specimens. So no one who works in botany knows you really need type specimens of a species name that are linked to a name, can really help decipher exactly what the author of that name meant when he described it. So he's like, oh, this is a species. And when you don't really have very well preserved type material, or sometimes they'll use images a lot, iconotypes, because it wasn't possible to preserve a physical specimen. And you can't put a picture under an SEM, or you can't turn it around. So the quality of the image is crucial for how useful it is. So there are a lot of problems. I think people have come up with techniques where you can preserve on a slide a particular cell, but I'm not sure how long they're supposed to last. You know, microscope slides are notorious for not being permanent. So yeah, long-winded answer to say it is a bit problematic.
No, that's a great detailed answer. In fact, I think all of us are so spoiled by those beautiful floral images in herbariums that it's difficult to imagine these in collections. Yeah. You have a question from Pauline Lang. Why are the chloroplasts so diverse in Desmids? Do we know?
I've no idea, really. They just are, aren't they? And it's amazing. And whether why they're particularly adapted. Why have such a complicated structure if you didn't need to have it? Because they can't angle themselves to catch the light. Things like the netrium, which you got up on the right there. I don't know. I mean, whether they're maximising light opportunities, considering they're, you know, often in, you know, they're kind of amongst pond weeds, and they might not be in the most brightest spot of the pond, but I can't really see why that would be such a major advantage, really. So I don't know. Interesting question.
Next up is Lawrence Carvalho. His question is, given their abundance in dystrophic waters, do we know their role in sea cycling in peatland landscapes? Can they utilize organic carbon as a carbon source?
I'm not sure, actually, but they're using such. I've got a feeling, which is based on nothing solid in particular, that they must be having quite a big role in carbon cycling, because they're so abundant, particularly in peat landscapes, and people focus on sphagnum for good reason. We think, well, these organisms are fairly, are going to be, doing a considerable amount of photosynthetic activity, and I think they will be taking carbon in and acting as a carbon source to some degree. But I don't think it's something people have really looked into very much. So I don't think there's been much research on on their impact with carbon cycling. So it's a good question, and it's something I've thought of myself. Actually, I've asked myself the same question and not really found much out there about that at the moment. But particularly in those kind of landscapes where they are quite, a dominant algal feature, I think it could be something that's quite an interesting angle.
Right. George Short has a question. Is there much known about how desmids fit into food webs? Does anything preferentially feed on them?
They will form a food source. I think the mucilage as well as because they do look quite juicy, don't they, these little green cells. But the mucilage that they produce, I have a feeling that may also be a significant element of food in the food webs. You will get bacteria that associate with that, those mucilaginous sheaths that some of these species produce around themselves. Often you get bacteria associated with those, and fungal hyphae as well. So you will get other organisms associated with that and that using that as a food source. I'm not sure, but it's quite a considerable mass of stuff that these things pump out. So I think as well as these green vegetative cells, you've also got, all this, mucus that they're pumping out as well, which could be another element in the food chain.
A question from Stella Sanford. Is much known about different mating types in desmids?
Mating types, not quite sure what she means. As I mentioned, they don't sexually reproduce a great deal. For some species, it's never been observed. And if they do sexually reproduce, it's often because there's an environmental stress. If the little pond's drying out, like when your house plant might just start flowering when it's about to die because you haven't watered it in 6 months, and there's a similar kick in if you get stressed, then that can often trigger sexual reproduction, but otherwise they don't really do it very often. I think it's just, it's quite a lot of bother. You've got to align, get close to another cell, turn yourself into an amoeboid state. So I think it is quite a hassle, and it's a lot easier just to reproduce vegetatively.
Question from Jan, or I don't know if it's Jan Kroc. Do you think the use of molecular tools would help in use of desmids as tools to assess W water quality? I'm guessing that is, or is that rare and they lack a DNA reference library make this difficult?
Yeah, well, I think yes. From what you've related, I think there could be really useful tools for indicating water quality. In fact, people, that's not new. People have been using desmids for assessing water quality, but the problem is trying to survey for these things when you're just restricted to, looking at one drop at a time of water under the microscope. So I think to try and improve the scale that you could do that work, I think it would be phenomenally useful and informative to be able to make use of more DNA tools. And as Jan points out, it's at the moment the lack of reference libraries is a bit of a stumbling block to be able to do a lot of that work. But the techniques are there, but there's so much light. You get so much DNA data back from your environmental sample that you do need to be able to do your fancy technical work on that to say, oh, this DNA is relating to this species. People are doing similar work in invertebrates at the moment, for example. So I have a colleague who works on things like mayflies, dragonflies. So you could have your dragonfly nymph, for example, and you suck its insides out or whatever of a specimen. take its insides out or a leg off or whatever you need and chop that up for your DNA work, and then you've still got a specimen left that you can reference. Okay, this is the morphology, so you've got a DNA reference, you've got a specimen that you can preserve nicely forever, and then you've got, you know, something that you can refer your data back to. But that's a bit problematic in the algae, as mentioned. Even having your preserved specimens can be a bit problematic. And also, ideally, you'd want to say, you know, I'm sure this is the same cell as what I have got a DNA reference for, but preserving single-celled and maintaining them as live cultures is you know, it's quite resource heavy, so it's not without issue. But I think that's, you know, that's kind of the direction that the research is going to go in, and there's a lot of really fascinating stuff that I think will come out of it.
Right. I'm sorry, we have a lot of questions. We'll probably keep you here all afternoon.
Oh, right,
I'm going to.
I might not know.
It's a question from Giuliano Morimoto. Do you think that the hypothesis-driven focus of research councils funding research is underpinning the lack of data on the natural history of desmids or other small organisms?
I think the lack of phycologists probably is impacting on the lack of data. Right. I mean, you know, I think there's probably a lot of reasons. We do know a lot about the species. It's the biological recording data so critical and useful for making those kind of assessments about conservation and the distribution and status. And that's often, you know, the work of many people. You know, it's often not, you know, a research grant that gets that kind of work in. It's having a group of skilled, dedicated people who go out and look for these things in ponds and lakes, as people do for flowering plants, bryophytes and lichens. And you know, we've got one person, Dave Williamson, who's been extremely productive and is a real expert on this group. And there are other groups, you know, people working on these, but it's very small compared to other groups. I mean, there's a fantastic effort in the Outer Hebrides, for example, with their biological recording group. There's a group going out recording desmids now and doing fantastic work, and so we've got some activity, but you've not got, you know, the same amount of data being collected as you would for other natural history groups. And perhaps one of the obvious reasons is they're quite difficult to identify. I mean, they're microscopic, which isn't necessarily a problem, but identifying the species can be quite tricky, because they can look very similar and have very minor differences. Often the genera are quite, I think, on the whole, can be quite easy to identify, but the species can be a little elusive sometimes. So you know, you need the literature and the support there to actually identify these things. Not sure if that's the question, but.
Question from Ingrid Jutner. Is there a sampling strategy in the Desmid community to cover habitats in undersampled regions of Britain and Ireland?
No. Short answer to Ingrid. I don't think there's that many people looking at desmids, really. I don't think there's as much of a desmid community as there might be a diatom community, and there isn't much of a diatom community either compared to other. Ingrid works on diatoms compared to other natural history groups, but I'm quite keen. Like I say, there are groups. I think you've got to be realistic in what we can achieve, and we're not, you know, perhaps targeting particular sites, as Ingrid alludes to there, targeting particular sites and trying to get some good quality data, and perhaps even trying to get some monitoring data as opposed to ad hoc recording data, to look what's happening at particular sites over time, is really valuable. I'm kind of hoping that we might be able to get a little bit more regular data from the site I was at yesterday, for example. It's probably the most important Desmid site in the southeast. So to be able to try and just get some regular sampling and a little bit more structured monitoring to see what's going on in those communities, I think would be really valuable. But that's probably what we need, as Ingrid suggests, and perhaps that's something that we can build up, and get our small community together.
Question from Michael Thane: can Desmids take up eDNA?
Well, eDNA is kind of just that's there in the environment. So I've not really tested this particularly, but I was wondering if the mucilage that they give out would have DNA within that. Because, for example, when a newt swims through the pond, as it sheds skin cells and mucus and stuff, these, you imagine your CSI forensic team where you pick off, bits of skin cells and stuff, and they do DNA profile in about 10 minutes in the lab. You can kind of get this trace DNA that's just discarded into the environment. I mean, but how effective that would be for these organisms, I'm not sure really. Something that'd be interesting to look into, I think, because we need there's so many amazing techniques. I mean, it's fantastic what we can do now with the molecular analysis and exploring that, molecular world. So it's amazing what we can do, and it's just trying to apply different techniques to different scenarios and see what works really. So.
Right. I know we're running out of time. Just going to take a couple of more questions because there's lots in the chat. Question from David Hill: how ubiquitous is distribution from the poles to the equator? Are they found in all habitats, or are there some that are more tolerant of different pollutants?
Yes, so I think they're pretty much, I'm not sure whether you'd find any in the Antarctic. I have a colleague, Anne, who works on Antarctic lakes. I don't think it's mainly cyanobacteria that she works on. So, but on the whole, they've got a fairly worldwide distribution. I think from what I've read, I get the impression that the centres of diversity for this group are more tropical, which I was a bit surprised at really, just because they're so diverse in peatland habitat, kind of North Scotland. But I think the suggestion I've read is that the diversity, hotspots and perhaps origin is more tropical. So they will be very diverse in tropical regions, but you will actually get these algae all over the world, really. I mean, they're fairly ubiquitous. They are restricted to fresh water. So as far as I know, there's always one example, isn't there, that bucks the trend. But as far as I know, they're restricted to freshwater environments. But that can include, as I mentioned with Desmids Ateniaceae, growing almost terrestrially, say over wet mosses, lichens, or a little bit of water running over a rock surface. So if there's a film of water, that will be enough for some groups of the Desmids. In fact, that might be preferred habitat for them, and they have all that gelatinous, that kind of gloopy goo to help with the desiccation. So there can be a whole range of habitats. For example, they're most predominant in a slightly acidic habitat, like sphagnum bog. But you can find, they'll pop up all over the place. Certain species are more widespread and happier in slightly more nutrient-rich environments than others. So you'll get species that are really restricted to these, oligotrophic, nutrient-poor habitats, and then you'll get some species that you'll find all over the place. I've did a little sampling in a pond down the road, large pond, ornamental pond down the road on the edge of Kent, for example. Wouldn't be a particularly, not exactly a classic Desmid habitat, but there was still, I found a few. There's some Closterium, a couple of Cosmarium species. So you will find certain species of these pretty much all over the place. And then if you go to one of the kind of prime habitat, then you'll get loads. When you squeeze your bit of sphagnum, a whole kind of green pea soup of Desmids will kind of drip out. I mean, that's the kind of peak Desmid, I suppose. But they will grow all over the place.
Okay, I think we can fit in one last question. I'm really sorry. I apologize for the rest of you. Emma Gray asks, do we know if the Desmid community changes with season? Could time of year determine what species you record in a site?
Yeah, that's a good question. They do change. There is a slight element of seasonality, but they will persist over winter. And but I think there is an element of seasonality to them. One thing I do wonder as well, though, is they're very associated. Like, for example, the Utricularia, that carnivorous pondweed that I mentioned, is a particularly good substrate, as it were, for the Desmids to grow on. So sampling a bit of that does, squeezing a bit of that plant into a jam jar and letting the water run off does. If there are Desmids around, they're usually on that. And that plant is seasonal, of course. It only appears in, where we are in UK, it appears, I don't know, May, June, and then disappears over winter. So there could be a kind of added layer of seasonality in terms of sampling bias. So the Desmids could still be there, but they're not as easy to sample, for example. That's just a suggestion. But she's right. Yeah, I think there is a certain seasonality as well, which you need to take into account for these things. And they're certainly most abundant in late summer, early autumn. So the numbers go up then. And because they're quite rare, I mean, they might, certain species might be very infrequent in a habitat. So if you've got them at their maximum populations, even though they may be persisting at some level all year round, you're more likely to sample them when they're frequent and in the prime of their time of the year. So so I think that's a good point, certainly.
Thank you so much, Jo, for your time and effort.